very much for the introduction. And thank you to the organizers of this meeting for giving me this opportunity. I'm Evelyn Jiagge. I'm a breast surgeon by training and currently a PhD candidate in the cancer biology program at the University of Michigan. The work I'm presenting here today is part of an international effort to find ways of bridging the disparity in breast cancer outcome. When you take two women with breast cancer, the same type of breast cancer, from different ethnicities, and you give them the same kind of treatment, the outcome is different between these two women. And we want to see why we are seeing ethnic differences in breast cancer outcomes. This slide is a slide that maybe many of us have seen before. It is the CS program that shows the incidence and mortality of breast cancer from the year 1973 all the way to 2007. Initially, the white represent the Caucasian population, and the black represent the African American population. The incidence of breast cancer in the whites is higher, and it's consistently remained higher over the years. But the mortality, as we are seeing here, was the same between African Americans and Caucasians, but now it is lower in the Caucasians as compared to the African Americans. And that was it started from the year 1983 with the introduction of tamoxifen, which is a targeted therapy for breast cancers that express the estrogen receptor. This morning, Dr. Robin introduced us to the subtyping of breast cancer. When you take all forms of breast cancers, they can be subtyped based on the expression of receptors on their surface, either the estrogen receptor, the progesterone receptor, or HER2. And if any tumor does not express any of these markers, it is referred to as triple negative. So those groups of tumors are referred to by what they do not express and not what they express. So these triple negative breast cancers, as we were told this morning, tend to be very aggressive and of high grade. They tend to have high cell proliferation, and generally they have the poorest outcome of all breast cancers. There has been a study that shows that the incidence of these triple negative breast cancers are very high in African American women here in the US. And the incidence, the slide, is up to it's 26% in African Americans as compared to 16% in white women here in America. There has been other studies that has confirmed this study. This is like the incidence of triple negative breast cancer in African American and in Caucasian. It can be as high as 46% in African Americans and as low as 8% in Caucasians. And these are the cancers that has the worst outcome of all breast cancers. In Africa, it has been shown that the incidence can be as high as 82% in an institutional-based study that was done with the University of Michigan in Ghana. So the question, the goal of our research is to understand the biology and the genetics of breast cancers that are more prevalent among women with African ancestry, and to create a panel of mouse xenograft models based on these triple negative breast cancers that can be used for studies. The rationale here is that triple negative breast cancers, we use women from Ghana because if we want to understand what is going on in African American women, or women with African ancestry, we have to find women with pure African ancestry. And since the slave trade was carried mainly through Ghana, we thought understanding what happens in Ghanaian women will help us to understand what happens in African American women across the world. So we travel to Ghana, we operate on the women, and we bring some of the tumors from there on dry ice and implant them in mice so that we grow xenografts from there. And we also have a cohort of African American xenografts and Caucasian xenografts here for comparison for the work we are doing here. Previously, I'll be introducing the concept of the breast cancer stem cell, but what made us interested in looking at the stem cell compartment in these tumors was because we did a panel of staining, a panel of tumors from African women. So from Ghanaian women from Ghana, and from some patient African American women from Hereford, and some from Europe, and some other parts of the world. And consistent with results that was seen in Uganda, we realized that in 69 patients, Ghanaian patients who had benign tumor, they had a very high expression of the ALDH marker. And the ALDH marker is a marker that is used to enrich for stem cells in breast cancers. So even in benign tumors in women with African ancestry, we realized that they had a very high expression of these stem cell markers. And so we wanted to understand what if the stem cell compartment in these tumors was contributing to why they were aggressive. And we know a lot about stem cells. So I wouldn't explain what stem cell is at this conference because I believe you all know what they do. But then what I want to say here is that there are some markers that enrich for stem cells in breast tumors. And these, when we have, if you take the whole tumor, there's a subpopulation of cells in that tumor that are known as the so-called stem cells that can be enriched for by the CD44 positive and the CD24 minus, ESA positive, or the ALDH positive. So these are two separate stem cell compartments that are found in the breast cancers. And we wanted to see that if we isolate these stem cell compartments as compared to the bulk tumor, are we going to see a difference in gene expression between them that might explain for why we are seeing these ethnic differences in breast cancer outcome. So our hypothesis is that there's a distinct subpopulation of rare cancer cells which exhibit distinct signaling pathway enrichment that may confer to triple negative breast cancers their metastatic and drug-resistant characteristics. So this shows that our experimental pipeline. We in the operating room, we take the tumor from the patient. We do so in Ghana, at the University of Michigan, Vananda, and Baylor Hospital. And this tumor is either we have the formalin fixed compartment of it. We take saliva from the patient at the same time as we take the tumor, and we take a normal DNA from the saliva for sequencing, as well as the tumor DNA and RNA. We do a whole exome sequencing as long as long insert, and then we sequence the saliva as well. Then at the same time, we put some of the tumor in mice to create xenografts, and when the tumors are 1 cm in diameter, we take those tumors and then we dissociate them using flow cytometry. We sort for the stem cell population, which is usually about 1% of all the whole tumor. So we sort for the two separate stem cell populations, the ALDH positive, the CD44 positive, 24 minus ESA positive, and then the bulk tumor. So from each tumor, we have 3 different samples, and then we sequence this. We sequence these cells that we have sorted for. So for now, we have sequenced 15 different tumors, which is 45 different samples. And then we do a comparison, comparing between ethnicities and between tumor types. And so for our results so far, for the sake of time, we realized that the stem cells, the different subpopulations of stem cells, that is the ALDH, this is PCA, it shows like the global expression of the tumor. I don't know if you are familiar with PCA analysis. So every dot here represents one sample. And so it shows the global expression using all the different genes that are being expressed in the tumor to separate them, to see those that are similar will cluster together and those that are separate will, are not similar will separate out. So the red represents one subpopulation and the black another subpopulation. We realize that the ALDH subpopulation is very different from the CD44 positive, CD24 minus subpopulation. And in using their gene expression between these two populations too are very different. Then we wanted to see that would the stem cells separate these tumors based on ethnicity. So we wanted to see that do tumors from people with the same ethnic background, do they separate based on their ethnicity? At this point, they are beginning to separate out, but it is not very obvious that these tumors at the stem cell level are separated based on their ethnicities. And the same both for the ALDH positive and the CD24 positive, 44 minus. But then we realized that there are distinct pathways that separate these two subpopulations of stem cells. And these are the pathways that came up as being very significant between the two subpopulations of stem cells. There's an enormous work on these pathways that there wouldn't be time to present, that are showing, giving us reason to believe that they contribute to why some of these tumors are more aggressive than others. And one pathway that we are concentrating right now on is the TGF-beta pathway. When we followed up these one, we did Western blotting comparing the ALDH population in the CD44 positive and the and the ALDH. I mean, there has been various publications saying that these two are either epithelial or mesenchymal states of the stem cells. But then when we did the pathway analysis, there are very interesting results that are beginning to come up to show that these two are very distinct populations that might be contributing to why we are seeing these ethnic differences. In conclusion, it is in very aggressive triple-negative breast cancers, the ALDH subpopulation of stem cells are less proliferative from what we have seen so far. They may therefore contribute to resistance to chemotherapy. And then they have the activation of EC1 and 2 in cytokine signaling, and that may be an important contributor to their tumor aggression. We want to study the signaling of significant pathways in the ALDH subpopulation and determine if this stem cell subpopulation is less proliferative and thus are more quiescent and responsible for chemo resistance. There are a lot of people contributed to this work here in the US and in Ghana. Now I'd like to say thank you to all of them. Thank you.